All right, so I think we can go ahead and start. Hi, everyone. My name is Cristina Fernandez, and I'm one of the educators at the DNA Learning Center. Welcome to our November Meet a Scientist talk. I'm very happy to have Dr. Doreen Ware with me today. But before we begin, I would like to share some information about our DNALC programs. Here is our DNALC webpage at dnalc.chshl.edu, which has information about our school field trips and public programming, such as our Meet a Scientist talk series. And I want to remind you guys that there is one more talk coming at the end of the year. Dr. Chris Baker is going to be giving a talk about his journey, and you can already register. But I want to mention that we already have a bunch of talks prepared for the rest of the year, as you can see here. So when they will be available, just please join us and register for all of them. I would like to also give you a little bit of information about two of our new DNALC centers, the Regeneron DNLC at Regeneron Pharmaceuticals in Sleepy Hollow, and the DNLC in New York City at the New York City College of Technology in Brooklyn, downtown. We encourage you guys to attend upcoming open houses for these facilities, to tour the spaces, to meet the educators, and to learn about the available programming. And additionally, I have something kind of, I think, interesting for a lot of people. We are building a gallery within the exhibit at the space in New York City, which will contain portraits of the diverse community in New York City. So if you like photography and you are over the age of 18, you are welcome to submit photos that reflect the heritage of New Yorkers that live in one of the 5 boroughs. And you can actually get your photograph featured in the exhibit and win one of the $4,500 prizes that we have. So you are welcome also to share this information with anybody that might be interested. And today we have the pleasure to have Dr. Doreen Ware with us. She is a professor at the Plant Group at Cold Spring Harbor Laboratory. And she is also working for the Agricultural Research Services at the US Department of Agriculture. And she's going to talk about her life as a scientist and her work. So welcome to share that with us. And I want to mention, guys, at the bottom, you have the Q&A section. So if you have any questions throughout the presentation, you can put all those questions there. And then at the end, Dr. Ware is going to answer them. All right, so thank you all for being here. Dr. Ware, this is all yours.
As Chris said, I'm actually a scientist. I'm actually a government scientist that works for the United States Department of Agriculture, and I'm actually stationed here at Cold Spring Harbor. And working for the Department of Agriculture, I'm interested in really addressing some of the basic science problems for agriculture. You know, we have a balancing act between what we need to do to feed people and what we need to do to protect the earth and deal with that in a sustainable way. And so part of the things that my group is doing and my science is really about understanding what's in a plant genome so that we can understand those functions and potentially help improve germplasm to deal with some of these challenges. And so I really think about that as exploring the genome with an eye towards breeding and management. And to me, it's really exciting because I think, you know, plants are really different. Plants are in one location. They have to deal with their environment, and they have to be able to think about strategies to handle that. And one of the things that I am going to be talking to you about is how the plant genomes do that and their fluid nature. But when we're looking at these genomes, you should be able to look at that genome and then with the environmental contributions, be able to predict what it might look like later on. And so from a breeder's perspective, they think of those as complex traits like yield. So whether you're looking at that ear, or you're looking at the amount of biomass on that field, or you're looking at a quality trait like wine, those are the things that we're looking at being able to predict from the genome. Now, plants, I think, are really cool, really, really cool. So I don't know if many of you know this, but many plants are polyploids. In fact, polyploids are a driving force of evolution, and most plants have a history of, at some point in time, one of their ancestors was polyploid. And you get a polyploid two ways. You can actually duplicate the same genome, or you can have two genomes come together. And I'm going to be telling you some of the work that I've been doing with maize, okay, with working with the corn genome. So I study these genomes. I try to understand what are the functions of the genes in the genomes. I also know that by looking at those genomes, I can tell a little bit of some things about the genome's life history. And so my group has been working on the corn genome for a very long time. In fact, the project that I worked on with the corn genome was started in September of 2005. My son here was born in November of 2005. So there was quite a journey. And there's been a lot of time working on that. And the genome is now 12 years old. But that first genome, we could see a lot of different things. And that's what I want to tell you. And this is the part that I think is really as exciting as a scientist. I always feel when I go to work every day, it's like figuring out a puzzle. So when you look at maize right now, what you're looking at is a picture of the maize's chromosomes here. And so you see this is chromosome 6 here, and this is chromosome 4. And when we look at this genome the first time, we could see that maize was two genomes that came together. The genes that were lost, we can tell how maize, and the genes that were kept, we can tell how maize is adapting. And it turns out that what maize ended up keeping were these genes that do a lot of regulation of either the DNA or they do regulation of proteins. And so the genome was pretty smart about keeping what I want to call the middle managers. It also turns out that in grasses, we have a really high level of gene order. And what you see in this map here is these gray bars really represent those two ancestral, what we would call homologous portions of the chromosomes that represent the ancestral chromosomes. And these bars here, these first red one, the first bar here and the second bar represent the rice genome and the sorghum genome. And where there are colored blocks there, that represents where the genes are all in the same order. So the structure of these genomes are hold up over time with having a high order. Now, I told you maize was an ancient tetraploid where these two genomes came together. And then they came together, and then there was a lot of breakage and gene loss. And then we ended up again with about 10 chromosomes. And we could reconstruct what each one of those parents looked like. And by looking in the genome, we could tell that one of the ancestral parents had more of the genome in the current version of maize than the other. And so we call this the strong or the bully genome versus the weak genome that is losing space here. And this is really important because it tells us that these genomes are evolving. They have a history. They can continue to track their history. And there's interactions in such a way that one genome is still dominating over the other, even to this day. And when we looked at the genome and we looked at what it lost, we could see that genes were lost in big clusters, not just one at a time. And that was interesting. We could all do that by looking at the genome. Now, it turns out that, for me, it's very exciting because we as humans have been putting a lot of pressure on plants. We've been domesticating them, okay? And we've been selecting for traits. And so here you'll see the progenitor of the modern corn, the modern ear corn maize is teosinte. And that teosinte plant, during the domestication process, it went from this little tiny ear here and this bushy plant to larger ears, very upright plant and then a very large ear. And humans selected on that the whole time. Now, it turns out that I think of this as biology-enabled agriculture, because in this case, we actually have been able to understand what are the genes that are regulating some of these traits. And in this case, we know that one of the major contributors to the change in the ear was a transcription factor. And that's really cool for me, because it turns out that, when we look at a lot of these traits, it's all about changing slightly when and where genes are expressed. And a transcription factor is like a middle manager who can change many genes' jobs, just slightly tweaking them. And so it's pretty cool to see from my perspective that there is this transcription factor that we selected for, and that is contributing to the changes that we see in ear structure. So I started out from an agriculture perspective, we think about plant breeding evolution. And this slide is a courtesy of my colleague, Dr. Ed Buckler, who's also part of USDA. He thinks about it, we as humans have been selecting over the last 10,000 years for these visible traits. And we've been looking at the different species that we have been choosing to domesticate. Most of them have been for food, animals and plants. And what's happened over the last 100 years is that there's been the opportunity to look at more controlled trials, quantitative traits. And then with harvesting and things, that we can do more calculations. And so we've been able to refine those phenotyping a little bit more than what we did over the domestication process, where people were just selecting the best ear, for instance. So what we've learned is that from over the last 15 years with access to sequencing technology, it's really opened up things. And what we understand that corn genomes are very different from each other. They're different at the single nucleotide level, little small differences. They're different in their gene content, and I'm going to tell you more. And this is like really amazing. And this is part of the strategies that are a success for plants. They can actually copy their parts list, okay? And then they can make new parts. And by doing that, they can either have twice as many parts or they can change their parts. They also basically interact really well with what's called the jumping genes, or the transposable elements, that's associated with modifying the regulatory sequence between genes. And that can also change where and when genes are expressed. So what's happened over the last 15 years is all of a sudden we've really been able to get now many different genetic markers, or those single nucleotide variations, where we could track differences in ways. So think about in the past, you track things, you selected the bigger plant, but now we can track things that have a specific genetic component. And that's now really enabled what we call marker-assisted breeding, where you're able to track a SNP or track the genetic variation associated, for instance, with a large ear. But then when you're doing that, you're picking genes of major effect and you're bringing them along. What they've also been able to do, now that we have all these genetic markers available, is we can now use machine learning approaches. And this is where we basically feed information of what's in the genome, and we say this population in this environment really has the traits we want. If I give you a new set of germplasm that has a different genetic background, can you tell me which one is going to be most predictive of having the best outcome? And we do that by basically using these machine learning approaches that basically look across the genome and can identify regions of the genome, the whole genome, that are likely to have an outcome of that trait in that environment. So, and it's been the ability to have these markers. So for me as a scientist, it's been an amazing time, because I've gone from building resources like genomes, I've told you I've been working on this for a very long time, to now having an incredible set of resources to now do some functional work. But what I really wanted to tell you is more about these fluid genomes. So we now have this concept that not every genome has some differences. And we know that in maize, there is a big difference about the content. But it's only been this last year that we've really been able to look at that. So this has been a very exciting time. So there's this concept that if you look at genomes across different individuals, so for instance, different corn lines, that there will be a part of the genes that are the same and a part of the genes that are different, that are separate parts list here. And the parts listed the same, we would call the core part. The parts list that are somewhat common between those, they call the dispensable, and then they have the unique part list. All right, so we have that part list that we would call the core and the dispensable part. So I'm going to show you an example where in maize, what this corn plant has done on the left-hand side, it now has 3 genes instead of 2. So it has more expression of a transporter gene that makes it possible for it to grow well on acidic soil. So the only difference is between the lines here and the lines here is 1 gene. So plants have these very fluid genomes where they can make copies of things. We would have called this a private gene because it's just in this one line. And what's really exciting when you look at this is what people call the dispensable part, I like to call the adaptive or the responsive part. So when I look at these corn genomes, and I'm going to tell you a little bit more, and the data that we're looking at, I'm going to be showing you, really has just come out in the last year. When I look at the makeup between different lines, they share a certain percentage of the genes, and then genes that are separate. They also share a certain amount of the transposable elements, and some of those are separate as well. So what's happened in the last 3 or 4 years is the technology has come along that we can now sequence many genomes. So we've sequenced these 26 genomes here, and you can see there's differences in each one of the years. There's other differences about these plants. And we're going to look, these are supposed to represent about 95% of the genetic diversity that's segregating it as a reasonable frequency across different corn lines. So when we looked at each one of these genomes, this is the first one, B73, that was done here. And what you find is that almost all the genomes have about 39 to 40,000 genes. And what's really interesting down here is the ones in green are the ones that are maize specific. The ones in yellow are shared between sorghum and maize. And these ones here in purple are shared among other grasses. And these ones in red and pink are shared among all plants. They're very old. Now, when we looked in this, I told you about the core genes. It turns out that only about 30% of the genes are core genes when we looked across all those 26 lines. In fact, most of them came in that dispensable and private part. And we were able to see that still, remember that red gene or that bully gene, it definitely, after we looked at this, still contained the bulk of the genome of the genes compared to the other genome. But then we find that there are sets of genes that aren't in the spots where we would have expected them to be. Now, here's a nice thing that shows is we add in each one of those genomes, how many new genes do we get? So it turns out across those 26 lines, we had 100,000 genes. Okay, remember I told you there was only about 39,000 genes in each one of those corn lines. There's 100,000 across the 26. And you can see as you add each genome in, how many of them increase. Well, so now we think about it. People have known about heterosis for a long time. And George Shull, who was here at Cold Spring Harbor, this is an old blackboard showing some of the studies where he was looking at some of these things. Now think about if he had understood that each one of these lines only was sharing about 50 to 60% of the gene space. Okay, and so that when he's looking at differences here, he's seeing the interaction of the combination now of more genes coming together. I mean, this is pretty cool, and we now understand that. So when we're looking at these genes right now, and we look at maize, I want to compare that to what we see in human. Now, in human, studies from a long time ago showed, using SNPs, that the difference in diversity between a chimpanzee and a human was about 1.3%. At that same time, almost 10 years ago, we knew that the difference between two maize lines was more than human and chimp. In fact, when you look at this, human and chimp share a lot of genetic diversity, a lot, as compared to what we see of these maize lines here. And in fact, Barbara McClintock, who also received her Nobel Prize, who was here at Cold Spring Harbor and received her Nobel Prize for transposable elements, we know that they are a major contributor of this fluid portion of this genome here. In fact, the jumping genes contribute to a lot of the differences that we see between organisms' genomes. So here, if you look at plants, this is the range of the amount of transposable elements. And this is here what you would compare for a human. Now, if you look around, you can see a lot of examples of these, okay, in nature. And we do know that these transposable elements can also create changes in the upstream region of a gene, contributing to when and where a gene might be expressed. So, so one of the things that we talk about is, if you're at a wine party, you could actually say, give me the Cabernet. I want the Cabernet without the transposable element in it. Okay, because we know that the differences between these here is the insertion of this transposable element. Well, it turns out that plants, compared to vertebrate genomes, I'm showing you here mouse and human, and if you see the yellow bar, these are the size of the genomes. And you can see that corn, mouse, and human have a pretty similar size genome. But if you look at the introns that are in red, you can see that those introns are very different sizes, okay? The exon structures in blue are all relatively same, but you can see there is definitely a difference in the intron size. In plants, when you have an increase in genome size, you see a slight increase in the introns relative to each other. Okay, now why this is important is because these genomes are living with their transposable elements, right? They want to compact and they want to put all their information of a gene in a more compact region. So it allows the transposons to be able to hop around and give them some new genetic diversity. Okay, so this is where plant genomes are very fluid. So, what we've learned over the last few years is that these genomes actually have ways of just duplicating the genomes by polyploidy. So they have more gene sets and they rearrange, and this leads to speciation. These genomes can copy and make copies of genes to be able to reuse that, and they can set themselves up to be able to deal with an environmental change, for instance, and they can have an individual in the population that may have more success under those changes because they can't run away. And so their success as a species is to have individuals with a lot of diversity to be able to deal with changes and adaptation. Okay, so wild grapes. Wild grapes have come with baggage that they shed during domestication, okay? They really don't taste good, okay? But wild grapes have disease resistance genes. Now, disease resistance genes usually come in a flavor where they have copies that come together here, and these are representing a gene. So these are classes of genes that we see that the plant will make many copies of it, and they rapidly evolve. They're actually very similar to our MHC locus that is involved in our immune responses in human have very similar structures associated with rapid evolution of these genes. Now, what we've been able to do right now, and it's very exciting for me. So there was recently a threat of sugarcane aphids for sorghum. And here you can see a variety that isn't tolerant. And here's a variety of tolerant. Well, and here's the aphids that carry the disease. We were able to generate rapidly a genome. These genomes here have full chromosome arms. We had information of where the disease resistance gene was. We could look at these clusters here, and we could identify a locus that looked like it was in the region, refine that locus, and now we have a curated locus here. And this is a display of it. And what we found was there was 191 kb region that had 6 disease resistance genes in it. So now, we're able to rapidly go in and identify perhaps the functional units that might be involved in a trait that was potentially what we would have considered a private allele. So my group has been involved in the technologies to be able to make these good reference genomes. And my group is also involved in making the data about the genomes available to other scientists. And here's an example of a visualization that's part of one of our databases that we display. Now as a scientist, like I said, every day I go to work and I think, oh my gosh, this is going to be great. It's like solving a puzzle. I get to look at how these genomes evolve. I get to manage the data associated with those genomes. And I think the other part about being a scientist that's very exciting to me is I get to work with a lot of people. Okay. And so I get to interact with many other scientists, which I find very enjoyable and in different fields. So I interact with scientists who are biologists. I interact with computer scientists. I interact with data scientists. I interact with breeders. And so for me, it's very exciting, and I really enjoy interacting with people in different domains. Now, I don't know if you know this, but breeders have been mutagenizing seed for new genetic diversity for a very long time. And so here's an example where if you look at regular sorghum, and then this is an example of a mutagenized sorghum, you can see it has erect leaves. Now, erect leaves are great for growing more compact, more plants in the same field, right? And so this is a desirable trait. And I told you about the bushiness and moving it in teosinte to more upright. So breeders can introduce new genetic diversity by mutagenizing seed. And in this case, this was an EMS population. Now, I've actually worked with some of the breeders who've introduced new diversity into sorghum. And in that one line, very interestingly, all of the flowers are fertile, whereas in the normal sorghum varieties, not all of the flowers are fertile. The pedicel spikelets are not fertile. Okay, it turns out it's a single nucleotide change in one gene that leads to that difference. And in fact, that gene that we were looking at for one of the alleles, MSD1, is actually a TCP transcription factor. And I told you earlier on that in corn, one of the domestication genes were a TCP transcription factor. Turns out that in this case, that middle manager transcription factor is actually regulating genes in a plant hormone pathway. And it's the change of that plant hormone that leads to the differences in fertility. So in addition to crops, my group works with model plants for the genetics, and I work with Arabidopsis, and I've been working with Arabidopsis for almost 35 years. It's a long time I've been working with Arabidopsis. And I can use model systems to look at the networks, and then I can test which genes more rapidly in a model system are going to have an impact. So when I'm looking at a complex trait like nitrogen use efficiency that involves a lot of genes, I can first use a model like a Arabidopsis to identify candidate genes before I start thinking about how that would be translated to a crop. So what I want to leave you with is in this part of it is that we've moved in literally in the last 15 years, we are now doing what I call breeding 3.0. Actually, it's what Ed calls breeding 3.0. And we've also now have the opportunity with CRISPR, where if we understand the genome function, we can rapidly use that information for editing. And so we have now entered into what would be breeding 4.0. And so in the next few years, what we're going to see is many more crops having access to breeding 3.0 and 4.0. And that's been enabled by the technology innovations that are coming along. And also access to machine learning approaches to better understand these things. So I have a background. I did my undergraduate work at the University of California, San Diego. I was very interested in biochemistry, and I was also very interested in computer science. So I actually, in my undergraduate, I did my degree in plant biochemistry, and I also took classes in computer science. But at the time I was taking programming classes, I actually thought that the computer science was actually really boring, and I didn't want to spend time doing boring things, and I would rather work with the plants. I also, earlier on, when I was going to college, thought I wanted to be a physician. But while I was in college, I actually was an EMT, and I also worked as a nursing assistant, and decided that I really preferred the basic research and working with plants more than I preferred working with people. So I've had sort of a transitional path there. Also, I did my PhD at Ohio State. So any of you that are Buckeyes, okay, is Ohio State, where I finished my degree there in plant biology. And then at that point in time, okay, the programming became more interesting again because there was more data, there was more management of data. And then I proceeded to pick up the programming aspect again and the data management aspect again. And I combined that with what I would call my love of biology. And so for me, I had sort of a circuitous route of where I wanted to go as I was looking at developing my career. I know I really loved working with people. I thought I would like being a physician, but decided I did not like working with people that much that were unhealthy or dying. And I then pursued a more basic research, and I really like working in agriculture. I really like working in plant science. I think that the things that I'm doing now is going to have impact on how agriculture will become more sustainable in the future.